Hey guys, so today we're going to be talking about genetic engineering, and this is actually the last lesson of our genetics unit. So we'll have our unit test coming up, obviously, and then we only have one more unit left after this, and it's the shortest one, so it should all be smooth sailing from here on out. But let's get into our genetic engineering lesson. So we're going to be looking at what recombinant DNA is, as well as genetic transformations. We're going to talk about how restriction enzymes and restriction endonucleases work, as well as DNA ligase. We're going to talk about what sticky ends are and what the point of making sticky ends is. Then we're going to talk about what DNA sequencing is and how we do that. We're going to talk about microarray analysis, as well as phylogeny. So recombinant DNA is a combination of DNA that doesn't occur naturally. So it's created when scientists take a sequence of DNA from one DNA molecule and then move it into a different DNA molecule. So for instance, one that's commonly used is scientists have taken the gene that codes for insulin and added it to the DNA of bacteria, and what that does is it makes the bacteria produce insulin, and then we can take that insulin and give it to people with diabetes. Genetic transformation is when an organism is made to express a new trait by adding foreign DNA to its own. So for instance, like making glow-in-the-dark goldfish by adding like bioluminescent DNA. Well, the DNA that codes for bioluminescent proteins from a different species and adding it to the DNA of goldfish. Restriction enzymes are the tools that are used to create recombinant DNA and genetic transformations. So the restriction enzyme can go into the nucleus of a cell, and it can identify and isolate a segment of DNA. So it can identify a certain segment of DNA and essentially snip it out of the rest of the DNA molecule. The enzyme can find a specific gene sequence, and then it cuts along a specific gene sequence as well. And then we can take that DNA fragment and add it into another DNA molecule to create the recombinant DNA. Restriction endonucleases are just a type of restriction enzyme that cuts in the middle of a DNA molecule instead of just cutting the end off of one. Each type of restriction endonuclease or restriction enzyme all recognize a specific nucleotide sequence. That sequence is called the recognition site. So each restriction endonuclease can be used to cut different areas of the DNA depending on which nucleotide sequence they recognize. And we can predict where different restriction endonucleases will cut the DNA based on what their recognition sites are. Recognition sites are usually about 4 to 8 base pairs long and are usually characterized by complementary palindromic sequences. So what that means when we say that they're palindromic is that they would read the same backwards and forwards if you're reading kind of like the two different strands. So here we have C, T, T, A, 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 T, T, C, or G, 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 that kind of thing. The sticky ends are what we get from most endonucleases when they cut the DNA. So any restriction endonuclease that cuts in like a stagger, like you see up here, that creates a sticky end. These sticky ends are where the single row of nitrogenous bases are, where they're not still like paired together. So if we look at the previous slide, this first example creates sticky ends here and here. The fourth example has sticky ends here and here, and the fifth example has sticky ends here and here. These two, numbers 2 and 3, don't have sticky ends because, as you can see, these are straight cuts, and so we don't end up with any unpaired nucleotides. Also, just in case you're interested, most of these restriction enzymes are named after the bacteria that we steal them from. So E. coli is the restriction enzyme from E. coli, for instance. So yeah, so sticky ends are useful because if you put them next to their complementary base pairs, they will stick back together again by forming hydrogen bonds. So that's really useful when we're trying to create recombinant DNA. If we can create sticky ends on the molecule that we've cut out, so the little DNA segment, if we give it sticky ends and the DNA that we want to insert it into, we also give it some sticky ends, then those two pieces will like Velcro together. When restriction enzymes are being chosen, because there are a bunch of different types, first you want to make sure that the restriction enzyme that you pick is going to cut on either side of the gene that you want and not through the middle of the gene that you want. So you do need to know what the recognition sites for your restriction enzymes are. And you also have to have the sequence of the DNA that you're trying to get a chunk of. And then, of course, you usually also want a restriction enzyme that's going to create sticky ends and not those straight blunt ends. DNA ligase is something that we've already talked about back when we were going over DNA replication. DNA ligase is the thing that joins the Okazaki fragments, the little chunks of the lagging strand together. But when you have a chunk of DNA that you're removing using a restriction enzyme and you're putting it into another piece of DNA, you once again have to close the gaps between the original DNA and that new little chunk that you've put in there. And so DNA ligase is what does that. It kind of joins it all back together again, closes any gaps in the backbone, that kind of stuff. The complementary bases will naturally hydrogen bond with each other, but the backbone will not rebond, because that's a covalent bond, without the DNA ligase. That is a condensation reaction, by the way, that takes place. So a OH is taken from the sugar and an H is taken from the phosphate, and water is removed, and that's where the bond forms. So in this example here, it says that this sequence of DNA was digested using the endonuclease small, which recognizes the sequence CCCGGG and then cuts between the C and G. It wants to identify the locations where it would be cut. So CCCGGG, it would cut right here between the C and G on the top and the C and G on the bottom. And then it would also do the same over here between the G and C on the top and the G and C on the bottom. So how many fragments would you get? You would get 3. You would get either end plus the bigger one in the middle. And what type of ends does it produce? Well, because it's cutting straight through between the C and the G on both the top and the bottom, it would make blunt ends. You wouldn't get sticky ends from this. So when the foreign DNA is incorporated into the genome of a cell, and then that organism starts producing those proteins and exhibiting a new trait, that is genetic transformation. In order to get the new or the foreign DNA into an organism cell, you do have to use a vector, which moves the DNA in. One of the more common vectors that are used are viruses. Essentially, you take the viral DNA out of the virus, put the foreign DNA into the virus, and then the virus injects what it thinks is its own DNA into the cells. It's really injecting in the foreign DNA. The purpose of doing this is to give an organism new traits because it will start expressing that foreign DNA, and then we call that organism transgenic. So a genetic transformation makes a transgenic organism. And we make that using our DNA fragment. Plasmids are small little circular double-stranded DNA that we find in bacteria. A lot of the time we will incorporate the DNA fragment into a plasmid and then of course put it into a virus which will inject it into the cells for us. The plasmid has to be cut with the same restriction enzyme that the DNA fragment was cut with, so that the sticky ends will actually like match up with each other and stick together to create just like a bigger plasmid where you've inserted that new little foreign DNA piece. And then you'll take the plasmid and you can actually like replicate it a bunch of times so that you can make a bunch of recombinant DNA pieces. So this is kind of how that would work. So this kind of pink circle up here is our plasmid. We would cut the plasmid using one of our restriction endonucleases that makes sticky ends. You would then go into the DNA of another organism and cut out a segment of its DNA using the same restriction endonuclease, so you get the same sticky ends. And then when you take the fragment of DNA, you can put it into the plasmid and the sticky ends will stick together. GMOs, also called genetically modified organisms, are another name for organisms that we have genetically transformed in some ways. Many crops nowadays are genetically modified, whether to be resistant to pesticides or diseases or droughts and stuff like that. Corn and soy and all of that kind of stuff is incredibly genetically modified. We can also make food healthier or bigger or hardier. There is some concern that GMOs will interbreed with naturally occurring species and create like super weeds or animals that have lower survival rates. But it can also be good for the environment because if we can make genetically modified crops more common, it means that we don't have to use as much water. Because water is a limited resource here on Earth, fresh water particularly, that would be very beneficial. Gene therapy refers to the use of a vector, usually a modified virus, to introduce a fragment of foreign human DNA into a person's genome in order to treat a disease or disorder. This is most useful when you're treating a recessive genetic disorder. If you have a recessive genetic disease, in order to no longer have that disease, you just need one copy of the dominant allele. So if we can insert a copy of that dominant allele gene, that could be a way to treat someone with that type of disease. Other genetic diseases are caused because someone has a gene that just doesn't function properly. Again, if you put in a functioning version of that gene, that could also treat that disease. So there are lots of different genetic diseases and disorders that have the potential to be cured this way, though at this point only ones that are caused by only one gene. Could also treat cancer by inserting like an oncogene or a tumor suppressor gene or something like that. At this point, gene therapy is fairly dangerous. I mean, they've been doing tests on it and stuff since the 90s. But every now and then people do have like really terrible like immune reactions either to the vector or just to the foreign DNA, which can be deadly. So there have been people cured of like terminal illnesses with gene therapy. There's also been people who have died because of gene therapy. So it does still have a way to go, but it's definitely a promising thing. There are two different types of gene therapy. Somatic gene therapy would correct genetic disorders in somatic cells, so it would cure that patient. Germline gene therapy would correct mutations in germ cells, so the sperm and egg, and that means that that new recombinant DNA would be inherited. At the moment, germline gene therapy is banned in Canada as well as many other countries. And the argument for that is that a person can consent to having their own genes modified in some way, but your unborn offspring can't. And so since future generations can't consent to the genetic modification, then you can't have this germline gene therapy, which would affect your future generations. And then, like I said, gene therapy is still fairly risky, and because of that, it is also controversial. DNA sequencing is another type of genetic technology. Essentially, in order to use any recombinant DNA, in order to make any genetically modified organisms or transgenic organisms, you have to know the sequence of nucleotides for the gene that you want to work with. So DNA sequencing is just determining the order of all of the base pairs in a DNA fragment. DNA sequencing is also used for like the study of evolution to determine like the relatedness between different species genetically. It's also used in like forensic science to determine like DNA matches between people, like paternal tests, all that kind of stuff, paternity tests. There are two different methods that are often used to sequence DNA. The first is gel electrophoresis, and then the next is actually dyeing nucleotide bases. So gel electrophoresis is when you create a banding pattern called a DNA fingerprint that shows the order of different nucleotides in a segment of DNA. Essentially, you have this gel electrophoresis compartment apparatus thing. And so it is gel. It's like this gel substance. And there'll be 4 different columns within the gel. And each column will represent a different nitrogenous base. So like ATGC, for example. And a band will appear in the column based on where it is in the DNA. So you read from the bottom of the gel to the top, and the bottom is like the 5' end of the DNA, and the top is the 3' end of the DNA. And then you can compare the DNA fingerprints. If you have like 2 perfect matches, that means like you found your murderer or whatever. That means you like have matched the person's DNA to the person's DNA. More closely related fingerprints, the ones that have like more similarities in their banding pattern, means that individuals are more closely related. So this is what this would look like. So if we look at the example on the right-hand side, so we start reading from the bottom, and the lowermost band is in the C column. So it would be a C, and then an A, and then a G, and then a C, and then a G, and then an A, and then 2 T's, and then an A. So you just like read from the bottom upwards. If you were going to try the same thing with the example on the left, it would be like T, G, C, A, C, T, T, G, A, C. You get the idea. So yeah, you guys can practice these ones if you'd like. But yeah, that's all you do. You just read from the bottom upwards. And then dyed nucleotide bases is another way to sequence DNA. Essentially, what you're going to do is you'll take a segment of the DNA that you want to sequence and put it in a test tube. And then you'll also have like a solution that contains the 4 different nucleotide bases, so A, T, G, and C, each labeled a different color. So maybe in the solution, all of the A's are red, and all of the T's are blue, and the G's are green, or whatever. And then what you'll do is you'll put the solution with all of the different bases in the different color in with the DNA segment that you want to sequence, and you'll create like the colored bases will form complementary base pairs with the fragment that you put in. And so then the new strand that you just built on against the original strand is now all these different colors. And so if you see that the new strand is like red, blue, green, well, that means it's like A, T, G, and then you can work backwards. If you know that the new strand is ATG, well, then the original strand must have been TAC or whatever. They had to be like the complements of one another. So you just work backwards like that to isolate the original strand. Polymerase chain reaction, also called PCR, is also really important in any genetic study or like forensic science and stuff like that. Essentially, if you only have like one small fragment of DNA and you want to run tests on it, you don't want to like destroy that little fragment on the first test and then can't do any more tests on it. So you can take a DNA fragment and make billions of copies of it using PCR. Yeah. So what you do is you take your little DNA fragment and you heat it up to break the hydrogen bonds. So you like split the two strands of the DNA apart. And then each of those strands are used as templates. So you add in a bunch of nitrogenous bases, you add in some primers, you add in some DNA polymerase, and your two new strands each get a new complementary strand built on them. And then you just keep doing that again. So now you have two DNA molecules, you're going to split each of those in half, make new strands, and you have four. And so every time you do a PCR, you double the amount of fragments that you have. You're really just forcing DNA to replicate on your terms. There's also something called a microarray analysis, which involves opening up a cell and isolating its genetic contents and identifying all of the genes that are turned on. So if you want to know in someone's cell which genes are on and which genes are off, that's where the microarray analysis would come in. If a gene is turned on, it will make an mRNA, because if a gene is turned on, it is coding for a protein. And the only way to make protein is by first turning your DNA into mRNA and then your mRNA into the protein. So if a gene is turned on, there will be mRNA. So you'll have this microarray, which is a chip essentially, like a, I don't know, like a computer chip kind of thing. And it has thousands of these like microscopic little cells inside of the chip. And each one of those cells will have a nucleic sequence that can bind to specific mRNA molecules. And so you'll kind of open up the cell, dump all of the contents of the cell into this microarray, and then any of the mRNA that's present will bind to the microarray, and then you can work backwards to tell which genes are turned on based on which cells of the microarray end up with something bonded to them. This is really important when you're trying to tell if like a cell is functioning right. In cancerous cells, if you want to know like, why is this cell behaving this way? Why is it behaving cancerously? What genes have been turned on or turned off that's making it behave like this? If you want to know how different environments cause different genes to be expressed differently, all of that kind of stuff is why you would use a microarray analysis to figure out what is causing different genes to turn on and turn off in people. You may have heard of the Human Genome Project before. It used microarrays to locate genes responsible for thousands of different traits, diseases, and disorders. They found there are about 30,000 genes in the average human genome, which was actually way fewer than expected considering how much variety in human traits there are. But that did tell us that there's really complex relationships between genes, but a lot of traits involve a combination of environmental and genetic factors, stuff like that. But they did this so that we can diagnose illnesses faster and to also be able to determine if someone is like predisposed to different diseases. You may have heard of like the BRCA1 and BRCA2 genes before. Based on the Human Genome Project, they isolated these genes called BRCA1 and BRCA2 and realized that people with these genes turned on are way more at risk of developing breast cancer. So people who have had like mothers or grandmothers or sisters with breast cancer might want to figure out if they're at risk for that too and get tested and see if they have one of these genes that gives them that genetic predisposition. There's also a hope in the future that we can use something called pharmacogenomics, which is the idea that we can figure out what medications will work best for a person based on their genetics. Because there's a lot of different disorders and stuff that there's a variety of treatments for, and some of them work for some people and some of them don't. And right now we don't have a really solid understanding of why. Like when it comes to cancer treatments, there are some chemotherapies that work great for some people and nearly kill other people. And you know, it would be excellent if you get the chemotherapy right the first time before, you know, almost killing someone. So yeah, the Human Genome Project is really cool and yeah, very beneficial to humankind. And phylogeny we've talked about before in Bio 20. It's kind of like the study of the evolutionary history of different species or organisms. And we can use both microarray analyses and DNA sequencing to analyze the DNA of different species in order to determine the relatedness between species. We know that species that are more closely related evolutionarily will have more similar genomes. And like we talked about when we were going over this in Bio 20, it's way more accurate than using like looks or morphology alone to determine relatedness. Okay, the last thing I want to talk about is mitochondria and chloroplast. As you guys know, DNA is found in the nucleus of most cells, but DNA is also found in mitochondria and chloroplast. And it's hypothesized that this is because mitochondria and chloroplast used to be single-celled organisms before they became organelles, and that over time they evolved to become organelles. Essentially, the theory goes that they were eaten by a more complex organism that decided, instead of digesting them, to use them as power sources, because both the mitochondria and chloroplast give power to the cells. It's also why both mitochondria and chloroplast have two membranes, whereas no other organelle does. But anyway, I'm getting off topic. Mitochondrial DNA, also called mtDNA for short, is much more limited than your normal nuclear DNA. It's actually more similar in structure to bacterial DNA than the normal nucleic DNA. It's also identical to your mother. So unlike the DNA in your nucleus, which is a 50-50 split between DNA from your mom and dad, mitochondrial DNA is 100% from your mom. And the reason for that is that if you think back to the reproduction unit, all of the organelles in a zygote come from the egg. The sperm doesn't carry cytoplasm and organelles with it, only the egg does. So all of the mitochondria in the zygote, and therefore all of the mitochondria in you, come from your mother. So yeah, I always thought that was cool. So in mammals, we can actually like track maternal lineage using mitochondrial DNA. Yeah. Okay, that's it. That was a bit of a longer lesson. But like I said, it's the last one of this unit. So hopefully you enjoyed genetics.